Okay, so before we do anything new today, I want to review a little bit what we were talking about on Thursday, because there was a lot of information in that lecture. And I just want to go over the highlights again before we finish up the respiration chapter and then head into photosynthesis. So this slide is a picture from your book. It's an overview of all the reactions in cellular respiration that I'd like you to be aware of. So first step in cellular respiration is glycolysis. Glycolysis takes glucose, converts it into pyruvate, and in the process it converts a little bit of NAD plus to NADH and converts some ADP to ATP. So we had glucose to 2-pyruvate, and this had a net to NAD plus into net to NADH and to ATP or ADP to ATP. I guess really that should be net there. All right. So we've generated a little bit of ATP, but most of the energy from glucose is still in pyruvate. Some of it's been converted to NADH. There's a step that we kind of breezed over a little bit where pyruvate is converted into acetyl-CoA, two of those, and in that process you also make two NADHs. And then in the citric acid cycle, acetyl-CoA, so this is acetyl-CoA is further broken down, and it produces, well, technically 6 more NADH, 2 FADH2s, 2 ATP. So we've got another ATP here, and some more electron carriers. So again, glycolysis produces some NADH and some ATP. Pyruvate's then converted into acetyl-CoA, produces a little bit of NADH. And then acetyl-CoA goes through the citric acid cycle, and we produce some more electron carriers, NADH, FADH2, and a little bit of ATP. So at that point, all of those electron carriers, the NADH and the FADH2s, will all go to the electron transport chain. Okay. And the electron transport chain, we're going to generate a lot more ATP. So most of the ATP in cellular respiration is produced by the electron transport chain. So this is about 28 ATP is produced at this point. So, and keep in mind, before that we produced 2 in the citric acid cycle and 2 in glycolysis. So most of them are produced by the electron transport chain, and that requires the electron carriers to bring in the energy to support that. And I'm going to show you that again later in the next slide. So glycolysis, really followed by the citric acid cycle. Big take-home message, glycolysis and the citric acid cycle convert the energy from glucose into electron carriers, and then electron transport chain is going to take the energy from those electron carriers to make ATP. So really, if you remember nothing else about glycolysis and the citric acid cycle, you're stripping electrons off of glucose bit by bit to generate these electron carriers. And then, thing that I went into several times, and I definitely want you to understand how this works, is what's actually going on in the electron transport chain to make ATP. So these electron carriers, NADH, FADH2, those ones that had stripped the energy off of glucose, they will drop their electrons, which represents the stored energy, off at the electron transport chain. As those electrons pass through proteins that are embedded in the mitochondrial inner membrane, they drive active transport to pump hydrogen ions from the matrix of the mitochondria into the inner membrane space. So remember, our mitochondria has two membranes. This is happening right on this inner mitochondrial membrane, and you're taking hydrogen, H+, and pumping it here, creating a high concentration of H+. So you're creating this concentration gradient between the inner membrane space and the matrix here. So then those hydrogens will diffuse through ATP synthase. So because they're highly concentrated out here and they're not so highly concentrated in here, they are going to travel back through an enzyme called ATP synthase, which takes ADP and converts it into ATP. And that's our end goal. Our end goal was to generate lots of ATP, so that generates it, right? Okay. Things you were confused about when you reviewed this, or just hearing it now again, things from Thursday that you don't quite understand. Feel you have a good grasp of what's happening here? Not at all. So where does NADH and FADH2, where do they come from in general? So what are they? What's NADH? Yeah, it's an electron carrier. Where does it get the electrons from? Yeah, breaking down glucose, basically. So through glycolysis, citric acid cycle, electrons are stripped off of glucose, transferred to NADH, FADH2, and where do they transport those electrons to? Yep, the electron transport chain, right? So they bring them to the electron transport chain. Electron transport chain, those electrons are used to drive what? What is their energy used for? Well, they are energy. What is their energy used to do? What does the energy from the electrons transported by NADH and FADH2, what does it power? Okay, more directly. What's it power in the electron transport chain? So what is the electron transport chain doing? All right, how? So it makes ATP ultimately, which powers the cell. Okay. It's going to pump hydrogens. So most directly, those electrons power pumps to pump hydrogen across the membrane into the inner membrane space, right? Setting up a concentration gradient. So you're pumping ions from a low concentration to a high concentration. And then what's going to happen? Where is ATP made? So where is ATP made in this process? All right, ATP synthase is an enzyme that makes ATP, and what powers that? Well, ATP comes from ADP, but what is actually giving ATP synthase energy to make ATP from ADP? All right, the hydrogen diffusing through it. So hydrogen coming from the high concentration and flowing to the low concentration. So it's just like water rushing through a turbine in a dam. Hydrogen are highly concentrated in that space up there, and they're going to flow through ATP synthase. Okay, so this is about the level of detail that I expect you to know this at, that you know that glycolysis and the citric acid cycle produce a little bit of ATP, but they mostly produce electron carriers. Those electron carriers take electrons, dump them off the electron transport chain. Electron transport chain uses the energy from those electrons to pump hydrogens across a membrane, setting up concentration gradient. That concentration gradient drives ATP synthase to generate ATP as the hydrogen flow through it, right? Okay, so that's really the whole thing summarized into a pretty short little thing. That's your entire chapter, basically, right? Okay, so that mostly makes sense for most of you. Okay, if not, then you really need to review that a few times, right? Okay, so that's the basics of the electron transport chain. Okay, I'm going to come back to this picture in a second. Yep. Okay. So just a few more things, and there's actually one other aspect of cellular respiration that we haven't talked about yet that we will talk about. Of course, I've very, very much simplified this whole process. And we've been looking at cellular respiration, so the process that makes ATP, as if there's only one input, as if glucose is the only input. And we know that's not true. You know that you don't just eat a diet consisting of glucose by itself. It'd be fantastic if we could. We would just eat candy all day, right? And that'd be it. But we know that that's not how things work. That's not at all healthy, because we also get energy from other fuel sources as well. So remember we talked about major classes of biomolecules. We had your carbohydrates, polysaccharides, monosaccharides. We had fats, and we had proteins. All of those can feed in to this process. And by feed in means that they can all donate electrons. So the energy to drive the electron transport chain comes from electrons. So each of these different components of your diet can provide electrons, and they provide them at different places, and by no means do I expect you to know where they do. If you take nutrition, you might learn more about this, but a lot of them can feed electrons into glycolysis. Fats and proteins can also feed electrons directly into the citric acid cycle or acetyl-CoA. So they can all provide electrons by following various pathways that all end up at the electron transport chain. So we're not going to talk about any of those other pathways because it's confusing enough just learning this one pathway. But just know that this is one out of many different routes to get to the electron transport chain, which is where a lot of ATP is produced. So you can have many different sources of electrons from many different types of fuel, so fats and proteins as well. And this is why you don't just eat carbohydrates. You eat other things as well. And actually, fats and proteins often are metabolized when you use a more wide range of reactions than just carbohydrates. Okay. All right. So just going back to this slide briefly, just wanted to highlight something that we talked about kind of briefly in class on Thursday, which is where oxygen comes into play here. So right by where that skull is where oxygen plays a role in cellular respiration. So oxygen combines with hydrogen to make water with the addition of electrons that are coming off that electron transport chain. So oxygen is necessary to have a location for those electrons to end up. So if there was no oxygen present, what do you think would happen to the electron transport chain? All right, so it's going to stop producing ATP because those electrons are going to back up. If the electrons have nowhere to go, it's like, closing down a road. You now can no longer use that road. There's nowhere for those electrons to end up, so there's nowhere for the electrons to get off of that pathway, so the electrons can't take that pathway anymore. And if the electrons can't flow down the electron transport chain, those pumps can no longer pump hydrogen, and then eventually you're no longer going to have a concentration gradient and you're not going to generate ATP. So oxygen is vital for this process to work. Oxygen is the final resting place of the electrons. So another thing here, just to point out before we get into the next section, is to look at what happens to NADH when it drops off its electrons. NADH gets converted to NAD+. Okay? So keep that in mind. NADH is converted to NAD+. When it drops off electrons to the electron transport chain, those electrons flow through the electron transport chain and they end up binding with oxygen at the end there. So this is called aerobic respiration. This whole process that we've been going through, it's called A-E-R-O-B-I-C, so aerobic. So aerobic means with oxygen. So aerobic equals with oxygen, with O2, oxygen, okay? Okay, so there are organisms that can function without oxygen. Yeast is an example of them. A lot of bacteria can function without oxygen. Without oxygen, they have to use a process of respiration, cellular respiration, called anaerobic. So anaerobic is without oxygen, aerobic is with oxygen. Okay? Anaerobic respiration will be a mechanism for generating ATP when oxygen levels are either low or oxygen is absent. So because when oxygen levels are low or absent, the electron transport chain cannot produce ATP. These organisms that live in very low oxygen conditions need an alternate mechanism for generating ATP. Okay? So the first step here, glycolysis, which goes up through the production of pyruvate, glycolysis does not require oxygen to be present. Glucose can be converted into pyruvate, converts a little bit of ADP into a little bit of ATP. So glycolysis produces these two ATPs without oxygen being present. So organisms that do not have oxygen present can still do glycolysis. They can still take their glucose, convert it to pyruvate, and produce a little bit of ATP. What inputs do they need? What do they need to get this reaction to go forward? What do you see on this board here that has to be present for glycolysis to make ATP? So what are at the back ends of these arrows? Okay, NAD+, yep. ADP, and what else? What's the ultimate source of fuel? Glucose, okay, really. So simplifying it as much as possible, those three things need to be present to make any ATP at all. Glucose is the ultimate fuel source. If you don't have any glucose, you've got nothing to make ATP from. So let's assume that our organism that's without oxygen has plenty of glucose around. So they're not limited by glucose. They're not starving. They're just limited by their ability to do the electron transport. ADP, anyone know where that comes from? It plays a role in that. It's also an input for ATP synthesis. But what is ADP? Compared to ATP, what is ADP? You remember that from the chapter before this? Yeah, it's diphosphate versus triphosphate. So what happens when, so ATP is stored energy, right? What happens when you use up that energy? Give you a hint. That's the answer to my first question. So what happens when you burn the energy of ATP? What does it become? Well, you need more, yeah, but so ATP is the fully charged battery. What do you think ADP is? Less charge. So it's a discharge battery. So ATP is the full high energy form. ADP is the low energy form. So where do you think you get ADP from? Where do you get dead batteries from? Right, from burning ATP. So you get dead batteries from using up the energy of the charged battery. So ADP is a product of burning all your ATP. So if you don't have any ADP, what does that mean about what state it's in? You probably have a lot of ATP. So if you don't have any ADP, you probably don't need any more ATP because you got plenty of it, right? So if you don't have any dead batteries, you don't need to charge your batteries, right? So ADP is probably in plenty of supply. So the whole point of this, of me pulling teeth right now, is that this is going to be the limiting factor, NAD+. And do you remember one slide ago? Where did this comes from? NAD+. All right, it comes from NADH, and where does it get converted back to NAD+? The electron transport chain, right. So at the electron transport chain, this NADH will drop off its electrons, become NAD+ again, and the NAD+ will go back and be part of glycolysis, right? But without oxygen, we have no electron transport chain. So there's no way to generate NAD+, right? So this becomes limited. This is the problem for anaerobic organisms. Organisms without oxygen run out of NAD+. So a process called fermentation, what it's going to do is it's going to regenerate NAD+, allowing glycolysis to continue without oxygen. And here's what it looks like. The left side of this graphic is glycolysis. Glucose is converted to pyruvic acid, ADP is converted to ATP, NAD+ is converted to NADH. And what fermentation is going to do is take this pyruvate and convert it into something else. So here, this is lactic acid. And in the process, it's going to take your NADH and convert it to NAD+. Okay, so NAD+ there. And NAD+ will then go back and be part of glycolysis again. So fermentation regenerates NAD+, allowing glycolysis to continue producing ATP without oxygen present. Okay. So this is actually a reaction that humans do. When do you think humans might do this reaction? Have you heard of lactic acid before? Do you know when that might be produced? So under what condition does this happen? What's lacking? What's missing? All right, oxygen's missing. So do you know any time that oxygen might be missing in your life? Okay, yeah. Well, yeah. But when you're dead, you run out of glucose too. So that's part of the problem. You've got no reactions going on. But yeah. All right, yeah, during exercise. So let's say you go out for a jog. For some reason, you really want to go jogging, right? So you go out for a jog. And what happens when you start running? Okay, yeah, you start to sweat because you're hot. But what happens in terms of, what happens to your breathing rate? It goes up a little bit. Do you know why? All right, well, why does your heart rate and your breathing rate, both of them go up? But why is that? Okay. You're lacking, well, okay. All right, wait on that. You're jumping ahead of me first. But what are you lacking? What are your muscles demanding, do you think?
Oxygen.
Okay, why are they demanding oxygen?
Because you're using it up there fast.
All right, to do what?
To produce ATP.
Yeah, there we go. So your muscles are demanding ATP. So your energy demand goes up when you start running. You use a lot less energy sitting in class than you do going for a jog, right? So you go for a jog, your ATP demand goes up. So let's assume you ate a good breakfast and you got plenty of fuel in your bloodstream. What you're going to lack is you're going to need more oxygen to run that electron transport chain a little bit faster. So your heart rate goes up to bring oxygen to your muscles faster. Your breathing rate goes up to get more oxygen into your blood, right? Let's say you run a little bit faster. Your ATP demand goes up. What happens to your breathing and heart rate? Goes up as well, right? Eventually, you're going to reach something called your maximum heart rate, which is as fast as your heart can pump, and you're breathing as much as you possibly can. And this all depends on how fit you are, right? When you might reach that in 2 seconds if you're really unfit, or you might reach it a little bit later if you're a little bit more fit. But eventually you're going to reach a point where you can't get any more oxygen to your muscles. So what do you think happens at that point? So let's say you're in a race and you start running out of oxygen. What do you think is going to happen to you? All right, you're going to cramp up, or you're going to slow down, or you might have to stop pushing yourself as hard, right? So in which case you're going to lower your energy demand to where you are actually fulfilling that energy demand, right? Let's say you're being chased by a lion. Do you want to slow your energy demand by slowing down? No. You want to raise your energy production a little bit. So that all you need to do is get a little bit more ATP. And so what your muscles do is they switch over to anaerobic respiration. They don't really switch over. They just add this on top of the aerobic respiration. So you're producing as much ATP as possible through breathing and pumping your heart as fast as possible. But you need just a little bit more. So you're going to take some of your glucose and break it down without sending it on to the rest of aerobic respiration, so you can generate just a little bit more ATP. And when you do that, a byproduct is lactic acid, which makes your muscles really sore after a while. So you're not going to be able to do this for a long period of time, but it'll satisfy that demand maybe enough to get away from that lion before you get eaten, right? So athletes do this all the time. You push yourself so hard that your muscles start getting sore afterwards from going beyond what you're capable of doing aerobically. So if you've ever seen those charts in the gym that have a heart rate zone, and they say, this is the aerobic zone. Does that sound familiar? Anybody that's ever been to a gym that has one of those? The whole point of that is to keep your heart rate low enough so that you are still in that aerobic zone. You're not going into the anaerobic zone of exceeding your, the amount of energy you can produce through aerobic respiration, right? So, because lactic acid is not the best thing for your muscles. It's a toxin in there. And you're also not using all of these processes, so you're less likely to draw on things like fats and protein. So you're less likely to draw on those sources of electrons. Fats is probably the one most people want to burn a lot of, right? So you're really just burning the easy, simple carbohydrates that are in your bloodstream really fast. So that's the whole, when you hear aerobic workout versus versus an anaerobic workout, you want to keep your heart rate low enough so that you're not maxing it out. You have that buffer zone where you're able to stay in this full process. So this is one form of anaerobic respiration. Anaerobic respiration occurs when oxygen is limited, and the whole purpose of fermentation is to regenerate NAD+. So here's another form of fermentation. This is alcoholic fermentation. This is what yeast do, and other microorganisms, bacteria, can do this as well. It's pretty much the same thing. The only difference is that pyruvic acid, rather than being converted into lactate, lactic acid, is converted into ethyl alcohol. So this is what happens when beer and wine are made. This is what happens when you bake bread sometimes. So as you're taking glucose and ultimately converting it into ethyl alcohol, and the reason why the yeast have to do that is that they need to regenerate NAD+. So why do you think a brewer that's making beer, they have their vat, what they do, they make up this really sugary mixture. Basically, they extract the sugars from grains, and they have this big vat they call wort of just sugary water, and they throw in yeast. And then what they do is they put an airlock on the top, and the airlock lets gas escape, but it doesn't let any gas come in. What do you think they're trying to prevent from coming in during the fermentation process? Oxygen. Why would they do that? What would be the problem with having oxygen get into the yeast? Okay, if oxygen gets in, the yeast are going to take that glucose, convert it to pyruvate, convert it to acetyl-CoA, convert it through the citric acid cycle, go through the electron transport chain, and the product of electron transport chain eventually is ATP as well as water, right? They don't want to make water, they want to make ethyl alcohol, right? So they want to prevent oxygen from getting there so that pyruvate gets converted to ethyl alcohol rather than ultimately to water, right? So exactly, that's why you would do that, so they keep oxygen out. Okay? So fermentation occurs when oxygen levels are low. It regenerates NAD+, and that's really the ultimate for that. So with plenty of oxygen, electron transport chain functions much more efficient, produces a lot more ATP per molecule of glucose. So you've got 28, 29, 30, 31, 32 ATP per molecule of glucose, as opposed to anaerobic respiration, which only produces 2. So we wouldn't be able to survive on anaerobic respiration alone. That's why we only use it as a way to give us a little bit of a boost when oxygen is low. You can't hold your breath forever, right? Because you demand way more ATP than you can produce anaerobically. Yeast can hold their breath forever. They're very small, single cell. Even though they can do aerobic and anaerobic respiration, doesn't really matter to them. They both produce plenty of ATP per molecule of glucose for them to survive. So you can have yeast in anaerobic conditions for a long time. Okay, questions on this chapter? Yeah.
Is there any way to get alcohol from like yeast and like sugar without sealing out all the oxygen, or this won't work that way?
It'll be harder. I'm kind of simplifying things. There's many different strains of yeast, some which prefer anaerobic respiration, and some which are more likely to do aerobic respiration. So if you have a strain of yeast that really only do anaerobic respiration, it might be fine. Yeah, and that's the thing. Brewer's yeast is more prone to doing anaerobic respiration, and actually oxygen does other things besides just stop the process of anaerobic respiration. But you know, one thing that brewers do is they will oxygenate their sugary solution when they first they'll oxygenate their mash when they first put the yeast in, because the yeast will grow really fast, and then they seal out the oxygen so that they will definitely convert over to anaerobic respiration. So, but really, no, most yeast are going to do aerobic respiration if they can. They produce a lot of CO2 anyway, and usually it covers, kind of the wort, the mash becomes covered in CO2, and it prevents oxygen from getting in regardless. So there are other techniques. Yeah, there are other techniques of doing than having a lid on your container, but yeah. It's a neat process, and it's very biological. Right? So, all right, other questions? Okay. You guys feel pretty good about respiration? You just don't want to say anything? All right. Okay. So we're going to move on to Chapter 7, and we'll get a little ways through Chapter 7, and we'll finish up Chapter 7 on Thursday. So So chapter 7 is all about photosynthesis. So we're going to see how photosynthesis is related. There's a lot of overlaps between these two processes, a lot of overlaps between photosynthesis and respiration. There's a lot of similar concepts to them. So having a good understanding of respiration is going to help you a lot with understanding photosynthesis. So showed this graphic at the beginning of the cell respiration chapter. Cell respiration is on the bottom side of this graphic. It's taking glucose and oxygen and converting it into carbon dioxide and water, and in the process releasing energy as ATP. Photosynthesis is the top half of this graphic. Photosynthesis converts energy from sunlight into organic fuel. So carbon dioxide and water, the two molecules on the right side, are going to be combined with the energy from sunlight in chloroplasts here to create glucose and oxygen. So the formula, the chemical formula for photosynthesis is actually exactly the opposite of the chemical formula for cellular respiration. So it is, let me show you. So you've got 6CO2 plus 6H2O plus energy yields C6H12O6 plus 6O2. So this is the formula for cellular respiration. Formula for photosynthesis, which is exact opposite formula as for cellular respiration. It's not the exact opposite process, but it's the exact opposite formula. So what we're doing is ultimately taking electrons from water and sending them back to carbon dioxide. So remember, in respiration, we took electrons from glucose, transferred them to oxygen at the end of the electron transport chain. Here we're going to take electrons and protons from water and send them to carbon dioxide. When we take the H away from H2O, we're left with oxygen. And when we add an H to CO2, we end up with glucose, right? So that's the process that we're going to be looking at in this chapter. So lots of organisms do photosynthesis. It's not just trees, not just grasses. It's many different organisms. They include land plants, mostly land plants. There's some plants that are found in aquatic plants as well. Photosynthetic protists, so algae. So lots of different kinds of algae, red algae, green algae, brown algae. This is a kelp, it can be larger than a lot of trees, but underwater. And even microorganisms. So there are bacteria that photosynthesize. So photosynthesis occurs in plants, algae, and other protists, and some prokaryotes like bacteria. These organisms are going to be responsible for feeding not only themselves, but most of the living world. Without photosynthetic organisms, without organisms that completed this reaction, we would not exist. We can't generate energy from sunlight. We just can't do it. We need something to eat. We need something to get our energy from. And so humans either eat photosynthetic organisms directly, so we eat plants, we eat fruits and vegetables, or we eat things that eat fruits and vegetables, plants. Yeah. So we eat cow, we eat pig, we eat things like that. So ultimately, we're getting all of our sugars, all of the energy from these things originally. Okay. Photosynthesis occurs in an organelle called a chloroplast. So chloroplasts here's a leaf, here's a cross-section of a leaf, and then it kind of looks down inside of the cross-section of the leaf into one leaf cell. Each cell in a leaf might have 30 to 40 chloroplasts, and there are hundreds of cells in each leaf. And you think of, on a typical tree, how many leaves are in a typical tree. You've got millions and millions of chloroplasts in a single tree. Chloroplasts are very, very small. Within a chloroplast, there's several different regions, and in order to understand photosynthesis, we need to know a little bit about the geography within a chloroplast. So chloroplasts have an outer membrane, and inside the outer membrane are these stacks of little membranous sacs. Each one of these is called a thylakoid. That's really the only word I really want you to know is thylakoid. The fluid outside of the thylakoids here is called stroma. So I'm going to be talking about one process in this chapter that occurs on the thylakoid membrane. So that's on the surface of one of these stacks. And then I'm going to talk about another process that happens in the fluid inside the chloroplast. So it happens in the stroma. So you need to know those two words so you'll know where I'm talking about these different things from. So stroma is the fluid, thylakoid are the little sacs, membrane sacs. Okay. One more thing from this slide that I want to point out, because it will play a role near the end of this chapter when we get to it, is if you look at the graphic of the cross-section of the leaf, you'll see that there's a little hole on the bottom of the leaf where CO2 is going in and oxygen is coming out. That is a hole called a stomata. Stomata are on the bottom side of leaves, and they're openings that allow gas exchange. So they'll allow carbon dioxide to go in, they'll allow oxygen to come out. So leaves, which we won't get into, leaves are covered, most leaves are covered in a waxy coating that prevents water loss. And that's its real purpose, is to prevent water from leaving the plant. But it also prevents gas exchange. So they need somewhere where they can allow gas to go in and out. So just like us, we don't breathe through our skin, we breathe by sucking in air into our lungs. Plants breathe by sucking air in through their stomata. So that's the only spot of gas exchange. So we're going to eventually talk about stomata a little bit later on in this chapter. Okay. So that's a little bit of the geography, and we'll talk a little bit more about chloroplast as we get into it. So photosynthesis is going to use the energy from sunlight and the electrons from water to transform carbon dioxide into organic fuel. So that's what this reaction is, so just to kind of reiterate it. So light energy is captured to transfer electrons. It's the energy, it's the power here that's used to transfer electrons. So this is a reaction that requires energy input because glucose here is a higher energy compound than carbon dioxide. So it's going to have to add energy in. Photosynthesis is going to occur in two major steps. And we'll spend some time ultimately going into these. The two major steps that occurs, the first one is the step that captures the light energy. It's called the light reactions. And it will capture light energy and it'll produce these energy sources, NADPH and ATP. And you're going to see NADPH very similar to NADH from cellular respiration. It's an electron carrier. It's going to transfer energy from one reaction to another reaction. So the light reactions capture light energy. They're going to split water. They're going to produce oxygen. And they're going to make ATP and NADPH, so two energy sources. Those energy sources, ATP and NADPH, are then going to fuel a reaction called the Calvin cycle, which will convert carbon dioxide into sugar. So there's two steps. There's the energy capture step, the light reactions, and then there's the sugar building step, the Calvin cycle. So you capture light reactions, you build sugars in the Calvin cycle, and you're going to see the light reactions occur on the thylakoid membrane. The Calvin cycle occurs in the stroma outside of the thylakoids. So in the chloroplast, but in the fluid outside of the stroma. Okay. So we will break this down in a little bit more detail, but not terribly too much more in a little bit. But the first step of this process involves capturing light energy. So light hits the chloroplast. Somehow, the chloroplast has to absorb that light energy and apply it to fuel reactions, right? So before we go into the light reactions and talk about the details of how this happens, I want to spend a little bit of time talking about light energy and what happens to light energy. So light travels in waves. So light travels in waves. We can describe light or measure light by something called wavelength. And that's the distance between peaks of the waves. It's usually in a unit called a nanometer. So wavelength is the distance between the peaks of the waves. A wavelength of about 380 nanometers, if that wave hits your eyeball, your brain interprets it as the color purple. If something that has a wavelength of about 500 nanometers, if light of a wavelength of 500 nanometers hits your eye, you're going to interpret that as green. 700 nanometers, you interpret it as red. Things beyond 750 nanometers, so longer wavelengths than that, we can't see. Our brains can't interpret that light. Those are things like infrared, microwaves, radio waves. So that kind of energy, we can't see it. Things that are shorter than 380, UV, X-rays, and gamma rays, we can't see as well. Yeah. The sun has all wavelengths. So it's putting out light all the way from gamma rays and X-rays, all the way through radio waves. A lot of that energy is bounced off of our atmosphere. So only a small section of that actually comes to Earth. But all the wavelengths of visible light are in sunlight. So, yeah. If you want to know a lot more about this, physics class is going to tell you more than I can tell you. But that's basically the concept of light. Light is energy of a particular wavelength. If it's between 380 and 750 nanometers, which is actually a pretty small range of wavelengths, we can see it as a color. And outside that, we can't see as a color. Okay, so when light hits something, there's really two things that can happen. When light hits an object or a pigment, energy is either reflected or absorbed. So let's take this red marker, for instance. Red marker has a red cap. It has red pigment in the red cap. Light from the lights, which is essentially white light, it essentially has all the wavelengths of light, it might be missing a few, hits this red cap. Some of it is absorbed by the pigments in the red cap, and some of it bounces off the red cap and goes towards your eyes. What color light is reflected, bounces off the cap? All right, the red. Should make sense. You see red because red light bounces off the red cap and hits your eyes. Green marker, same thing, but the light that bounces off the green pigment is green light. What do you think happens to green light that hits the red cap? Yep, gets resorbed, exactly, because there's only two things. It can be reflected or transmitted, or it can be absorbed. Transmitted is just passes through it. So if you were to hold, if something's translucent, if you can hold it up to the light and some color passes through it, it's the same idea. The light is not being absorbed. So red light that hits the green marker is reflected as well. So we see the light that is reflected or transmitted. We do not see the light that is absorbed. So a leaf that is green, what light is being reflected? Green. Okay. This shouldn't be overly complex. I'm trying to very, very simplify it. Right? So what happens to blue light that hits a leaf, a green leaf? It's absorbed. Exactly. Purple light that hits a green leaf is absorbed. So every other color besides green is absorbed. And this is a picture here of a chloroplast. And there are pigments in the chloroplast, and we'll get to that, that will reflect green light, and they're going to absorb the other colors of light. Okay, so there is a tool that you can use to measure absorbance and transmittance in a lab. And basically what it does is it shines a whole spectrum of wavelengths on a sample and then measures how much is absorbed by the pigment. And you can end up drawing a diagram where you have wavelength, and it might be from 380 to 700 nanometers. So this might be, let's just say 350, 400, 450, 500. Something like this. And you're going to have absorbance over here. So if we look at an absorbance spectrum of a leaf, absorbance is going to be really high in this wavelength, be low here and high here. Right here, this is green. So absorbance is very low in the green wavelengths. It's very high in the purple and blue. It's also a little bit higher in the reds, but it's low in the greens. This is kind of what this diagram is. What this diagram is an experiment where light was run through a prism, separating it out into the spectrum of colors all the way from 400 to 700 nanometers. And that light was then shown onto an algal cell. So this on the bottom is a few cells of algae. So that only purple light hits here, only red light hits over there, and in the middle is green and yellow light. So what this is showing is bacteria that require oxygen growing on the algal cell. And when they measured the amount of bacteria growing in the different regions of the algal cell, they found that a lot of bacteria are growing in the purple part of the cell, not very much in the green, but more bacteria in the red. So what are the bacteria responding to, do you think? All right, so they're not doing anything with the light, but what do you think the algae is producing that the bacteria are consuming? What's a product of photosynthesis that you think bacteria that, well, you've got this and you've got this, right? So they're responding to one of those. They're probably responding to the presence of oxygen. They're able to survive in the presence of oxygen, whereas in the middle, there's no oxygen being produced. So what does that tell you about which color light drives photosynthesis really well? So does green light drive photosynthesis? Why not? All right, well, this is evidence of it. So there's no oxygen because green light doesn't drive photosynthesis. But why doesn't green light drive photosynthesis?
It's not absorbed.
Right, okay, so your chloroplasts, which look green, are not absorbing green light. So light that is reflected is not used to drive photosynthesis. And what this shows is that the light that is absorbed can be used to power photosynthesis. So what color light drives photosynthesis really well? Yeah, purple does really well. Red does okay. Blue does okay, right? But green, yellow here, orange doesn't do all that well. So purple light, plants love purple light. So if you were to shine just a purple light on a plant, it would survive just fine. If you shine just a green light on a plant, it's going to die. It's not going to be able to photosynthesize. Okay? So that's kind of the take home here. Oh, so I had a question here which we've already just talked about. Forgot I should have put this first. So what color of light do you think will drive photosynthesis by green plants most efficiently? Yeah. So green will not, right? Yellow will not. Red and blue both will, but when you have red and blue together, you're going to drive it the most efficiently. So how about this? This is a red alga. Why do you think this alga is red? So this is an aquatic plant. So why would it be red? Okay, there's the very simple answer, right? It reflects red light. So the reason why it reflects red light, what do you think happens to green light that hits it? Yeah, it's absorbed. So if it reflected green light, it would look green, right? But it looks red. It's because it actually, more so than it reflecting red light, it's absorbing more green light than a green plant does. And this is what the pigments look like in it. It has pigments that land plants have, chlorophyll A, chlorophyll B, and xanthophyll, beta-carotene there. But it also has phycoerythrin and phycocyanin, which absorb green and yellow light. And so all that's left, really, the only place where there's within the visible spectrum is right around 700 nanometers, which is red light. That's the only place where those bars are really low. So it looks red. The reason for this, have any of you been scuba diving? Have you been fairly deep?
Is it because the light won't reach down?
Yeah. So if you go scuba diving and you go, say, 100 feet deep, less so if you're further north, more so if you're further in the south, and you look around, everything looks kind of greenish, right? Unless you break out a flashlight and you shine a flashlight on things, and then they'll have all these crazy colors, right? And that's because green light actually travels really well in water. Red light doesn't travel too well in water. So a plant that is green is missing out on a lot of the energy that's getting that deep in the water. They're missing out on all that green light. The reason why things look green is because that's the light that's down there. It's bouncing off of things and making them look green. So red can be sacrificed because red light's not going to go very deep in the water, but green light travels a lot better. So, if you do go scuba diving, look for the seaweeds. Near the surface, there'll be some green seaweeds. Further down, there'll be some brown seaweeds. And deepest, there'll be red seaweeds. So you're going to see different color plants the deeper you go, and they're responding to changes in the light condition. All right? So the alga is red because it's absorbing green light and it's reflecting red light. And it's doing that so that it can absorb a lot of energy. So most plants are going to have multiple pigments in them, and each pigment is going to absorb light at a very specific wavelength. So this also goes to why trees up north especially will change color in the winter. What's happening is, although they have, so just imagine this graph without phycoerythrin and phycocyanin in there. They've got chlorophyll A, chlorophyll B, and beta-carotene. Chlorophyll A and B absorb violet and blue light, but they also absorb red light and some orange light. Beta-carotene absorbs violet and blue light. So in the north, over time, those chlorophylls are going to be hit with a lot of light energy. They're going to eventually break down. Plants are constantly replacing the chlorophyll that's in their leaves because as they get hit with light energy, they break apart. It's just like if you leave a colored t-shirt out in the sun, it eventually fades. Right? Over time, the pigments in that shirt fade away. Plants are the same way. If they didn't replace the chlorophyll, if they didn't replace the pigments, they would just fade over time. Up north, when it gets to be about October or so, end of September, early October, they stop replacing the chlorophyll because they're going to shut down their leaves and shed their leaves for the winter. So they don't want to keep making molecules that they're not going to need for very long. So what happens is the chlorophyll degrades and fades away. Beta-carotene there is a much sturdier molecule, and it doesn't fade as fast as the chlorophylls. So chlorophyll, which was absorbing yellow and red light, leaves the plant, and yellow and red light start being reflected. So if chlorophyll A disappears first, the leaf is going to look red, because red light will be reflected rather than absorbed. If chlorophyll B leaves first, the leaf might look yellow or orange. And then once both of them leave, the plant's going to start looking more of like a brownish color. It's going to start reflecting green and yellow and orange and red, and it'll eventually turn brown. So, and then it falls off. So if you ever wonder why leaves change color, it's because some of the pigments disappear and they start reflecting light that they were previously absorbing. They're reflecting red, reflecting yellow. Okay? All right. Let me see where we are. Okay, I don't want to get too far into this, but I am going to start introducing this next section, and we will probably review a lot of the next few slides again next class. So now we know plants absorb light energy. They do that by having pigments. Pigments will absorb light energy of particular wavelengths. So how do they use that fact to generate energy to transfer electrons? So how are they going to make things like ATP and NADPH by absorbing light energy? Well, they do that using a special structure that's found on the thylakoid membrane in a chloroplast. So these are very, very, very small things, and there's many of them in each chloroplast. And remember, there's 30 to 50 chloroplasts per cell. There's hundreds of cells per leaf. There's thousands of leaves per tree. So there's a lot of these in there. There are these units called photosystems. Photosystems will absorb light energy using chlorophyll molecules. So these green dots here are chlorophyll pigment molecules bound to proteins in a structure called a photosystem. When light hits one of those chlorophyll molecules, as long as it's not green light, it's going to be absorbed. So if it's blue light or red light, it's going to be absorbed by those chlorophyll molecules. And what that does is it energizes that pigment. And there's several things that could happen when light energizes a pigment. It can just destroy the pigment, which would be bad, but that happens sometimes in plants. It could cause the pigment to heat up. So this is why if you wear dark-colored clothing, the reason why you feel warm in the sun is that all the pigments in your clothing are being hit by light energy, and they're absorbing that light energy. What do you think's happening to that light energy if you're wearing like a white t-shirt? Yeah, so the reason why it looks white is that energy hits those pigment molecules and bounces off. But if I wear a black shirt in the sun, the light energy hits those pigment molecules and gets converted into heat. So heat, damage, they're not good things for these chlorophyll molecules. So instead, what happens is they get energized and they play basically hot potato. They're going to take that energy and pass it off to a neighboring pigment molecule. So you see the light photon hitting the one green dot, and then the arrow, the yellow arrow, signifies that it's transferring that energy. So rather than getting damaged or heating up, it transfers that energy to a neighboring pigment molecule. And by doing this, it's going to funnel that energy into the very center of the photosystem. So all that energy that hits the outside pigments is eventually passed to the center. So, and it's not just happening one at a time. There's lots of photons of light. So a photon is just kind of a packet of light hitting many pigment molecules. It's all being funneled down to the central pigment molecule. So you've got the central pigment molecule, and you've got light hitting, funneling to the central pigment molecule, like that. So this central molecule is now being hit with a ton of light. It's a lot of light all being funneled down to it. And all that energy, there's so much energy that it causes the molecules within that pigment to lose an electron. So that's what this electron transfer is. So it's going to cause electrons to be transferred from the central chlorophyll pigment molecule to something called an electron acceptor. And so you've energized an electron and shot it loose from that pigment molecule. And that's the very first step in photosynthesis, is funneling energy to a central pigment molecule and exciting an electron in order to send that electron to a primary acceptor within the chlorophyll molecule. Okay. All right. I think what I'm going to do, I'm going to go through this slide, and then I might just end things there so we can start with this whole concept on the next class, because it can get a little bit big. But I'm going to do an overview of this process, and we'll come back to this again on Thursday. So this is a picture, again, from your book that really simplifies things down, but each of those big green dots is a photosystem. So just like what was on that last slide, each one of those green dots is one of those photosystems. So in each of those cases, light will hit the photosystem pigments, funnel energy down to a central pigment molecule, and excite an electron. So what happens in this first photosystem is that excited electron is going to power an electron transport chain. So what do you think that electron transport chain will produce, ultimately? Yeah, ATP. It's on there, but yeah. So just like in cellular respiration, just like in the mitochondria, electron transport chain is going to generate ATP. And how do you think that happens? So how did it happen in the mitochondria? How did an electron transport chain lead to ATP being produced?
What was the question?
So electron transport chain, how does it generate ATP? So how did that generate ATP in the mitochondria? ATP synthase. Yeah, you're getting that right. So, and what powered ATP synthase again? Hydrogen, right? The diffusion of hydrogen. So same thing's going to happen here, and we'll see that on Thursday. Electron transport chain pumps hydrogen across a membrane, leads to a concentration gradient, leads to ATP production through ATP synthase. So good. At least you're remembering some of the words. You're on the right track. Okay. Second photosystem here, this one that's called the NADPH producing photosystem, that excited electron is just going to be transferred to NADP, making it NADPH. So just like NAD became NADH, and NADH had an excited electron that it carried elsewhere, this second photosystem will use the energy it captured from sunlight to transfer an electron to NADPH. Okay, so the first photosystem transfers an electron to the electron transport chain to generate ATP. This NADPH producing photosystem captures light energy to energize an electron to make NADPH. Okay. So when the pigment molecules within a photosystem lose an electron, they need to pick up an electron from somewhere. This NADPH producing photosystem gets its electron from the electron transport chain. So the electron that just went through the electron transport chain to dump off or to make ATP, when it's used up all of its energy, it ends up at the reaction center chlorophyll of the NADPH producing photosystem to replace the electron that just got sent to NADPH. Okay, so that's where that one goes. The first one, this water splitting photosystem, gets its electron from water. So the electron is sent from that photosystem pigment up to the primary acceptor and then through the electron transport chain, and it replaces its missing electron by splitting water. So water is split in two, electrons go to the reaction center chlorophyll, and you've produced oxygen as well as hydrogen. So this is where oxygen comes into play. Oxygen is produced really at the first step of photosynthesis. As soon as light hits that reaction center chlorophyll, that chlorophyll is going to pull an electron off of water. Water is going to be split into hydrogen and oxygen. Okay, so on Thursday, we'll start here, and we'll actually talk in a little more detail about each of these steps, and you'll know all about photosynthesis. We'll also see kind of an overview of this again, and we'll go into some special cases of photosynthesis to finish it up. So I'm going to stop here so we don't get into too much detail today. and then we'll continue on Thursday.